Yes, ma'am.
Yes, ma'am.
Okay, children, yesterday in the last lecture we discussed about the excretory system, which includes all four parts. Then we studied the LS of kidney. Now we will be talking about the even we studied the structure of nephron. But there are basically two important types of nephrons. These types we will see and we'll continue with the next part. So there are two types of nephrons as per the location they are named. Now you studied the LS of kidney where we studied outermost part, that is cortex, then medullary region, medulla, and the innermost, that is pelvis. Okay, so whatever the nephrons which are present, that means spatially this loop of Henle which is present in the cortical region. So nephrons which are situated in the cortical region, they are known as cortical nephrons. Whereas in case of some of the nephrons, that loop of Henle is long and it continues in the medullary region. So that those nephrons are called as juxtamedullary nephrons. So cortical nephrons, here you can see this is the cortical region, this is the medullary region. When you take the LS of kidney, this is the cortical region where this Malpighian tubule along with PCT, DCT, and most of the part of loop of Henle that is included in the cortical region. So, in majority of nephrons, the loop of Henle is too short and extends only a very little into the medulla. Most of the part is present in the cortical region only, cortex only. So, such type of nephrons are called as cortical nephrons. Whereas the loop of Henle, when it is longer in size, length, then in that case, those nephrons where the loop of Henle is very long and runs deep into medulla, so those nephrons are called as juxtamedullary nephrons. Okay, so these are the two main types of nephrons. Now we'll talk about the urine formation. Now you keep your page open, that is 182, where 17.8, figure number 17.8, the process of urine formation is given. You just try to understand. I will repeat that part again and again for your understanding, but you keep that text open. Okay, so that I will see. Can you see the urine formation slide? Now, this is what the structure of nephron. Okay. In this structure of nephron, you have learned yesterday that this is the Malpighian corpuscle which contains this cup-shaped structure called as Bowman's capsule, whereas this cup-shaped region, that is Bowman's capsule, which contains the blood vessels, the network of blood vessels in it, that is called as glomerulus. So this is one part, and remaining entire, that is called as, that tubular part is called as the common name, that is renal tubule. Now this renal tubule, as we discussed yesterday, is divided into four parts. That is, when this Bowman's capsule, it enters the smaller part which becomes narrow, that is neck, and then it becomes broader, and near this neck or near this Bowman's capsule, this tubular part, that means the renal tubule, is highly coiled, and so this part is called as PCT. That means what? Proximal convoluted tubule. So this part which is highly coiled is called as PCT, which is near to this Bowman's capsule, and that is the first region. Then this extends, forming a hairpin, U-pin or U-turn like structure or hairpin like structure, that is called as loop of Henle, with the two oppositely directed limbs, one is the downwardly directed, that is descending limb, and the one which is upwardly directed, that is called as ascending limb. And then it continues in the DCT, that means distal convoluted tubule. Here also the tubular part becomes coiled, so this is distal convoluted tubule, which then enters into the collecting duct and then into the common duct. So these are the four parts of the renal tubule that we have learned yesterday. Now each and every part is having its own function. Now when we are talking about the process of urine formation, in that case it takes place in three steps. First step is called as glomerular filtration. The second step is selective reabsorption, and the third step is called as formation of urine. That So these are the three. Formation of urine, before that formation of urine, that tubular secretion. Now first is glomerular filtration, which takes place in the glomerular region, that means the Bowman's capsule. Second is selective reabsorption, which takes place in the PCT and loop of Henle. And the third, that is tubular secretion, that takes place in DCT and to some extent in CT. So these all four parts are involved for a specific function. Now how it takes place and what is the meaning of glomerular filtration or tubular secretion or selective absorption, that we will talk step by step. Now first step is glomerular filtration, which is considered to be the passive type of process. Now you try to understand what does it mean by passive and active. Passive means where energy is not required for the process. Automatically, easily, in the normal way, it takes place without utilization of ATP or energy, then that process is called as passive. And active means where energy in the form of ATP generally it is utilized, that is considered to be active type of process. Okay, so accordingly, glomerular filtration is considered to be passive type of process, where filtration, that means you know very easily the meaning of the filtration, where something is getting filtered, right? So what is the word glomerular? Where are the glomerulus present? They are present in the Bowman's capsule in this region, isn't it? So when how it takes place this in the glomerular filtration, you know that the blood vessel called as arteriole, which enters this Bowman's capsule, is having the greater diameter than the blood vessel or the arteriole which is going out of this capsule, Bowman's capsule. I told you this while explaining the structure of Bowman's capsule. You know why? The function or the reason behind it, that is the difference between the afferent which is entering the Bowman's capsule and the efferent, that is which is leaving the Bowman's capsule. That afferent and efferent arterioles are having the difference in their diameter, where which is entering afferent arteriole is with a greater diameter than the efferent which is leaving. That helps in creating a pressure called as hydrostatic pressure in this glomerular region, where all these capillaries, network of capillaries of the glomerulus, which we call it as glomerules or glomerulus, is present in this region. So because as the blood flow is coming with in the Bowman's capsule with a great force, but it is that efferent arteriole is having the less diameter, it cannot go with the same force or the pressure, so that it creates a pressure in this region, that is glomerular region, and that is called as hydrostatic pressure. And because of this pressure created, how it is created? Because of the difference between these two afferent and efferent arteriole diameter. Okay, and that's why whatever the pressure is created, it helps for the filtration. And because of the difference in the diameter, whatever the pressure is created, that is glomerular hydrostatic pressure, it helps in the filtration. And so these glomerulus which contain blood, these are all the network of blood capillaries, with very small diameter. Their walls are very thin walled, and because of that, what happens? Filtration takes place, and whatever the filtration takes place is called as glomerular filtration. That is the first step. That is also called as ultrafiltration. So in the formation of urine, this is the first step where the network of capillaries present in the glomerular region, because of the change in the diameter of afferent and efferent arteriole, hydrostatic pressure is created, which results in the which results in the filtration of these the blood present in these capillaries, and whatever is filtered, that is called as filtrate, which from this region, cup like region, that is the Bowman's cup region, that enters in the space which is present between the inner and outer layers of this Bowman's capsule, which we have learned. So inner is visceral layer, outer is the parietal layer, and the space present between the inner and outer layer, this is called as capsular space. That looks like a capsule, and this capsular space that shows that stores, or where in the capsular space this filtered part, that means filtrate, that comes, and through this neck region it enters the PCT. So try to understand this. This is one more diagram where afferent arteriole, here is the glomerular region. Because of the hydrostatic pressure created, the filter that it from this glomerular part, it enters in the PCT, and this takes place passively without utilization of energy. So this is considered to be the passive process. So glomerular filtration is non-energy consuming process, so that's why this is considered to be passive type of process. So the first step in the formation of urine is the filtration of blood from glomerulus into the lumen of Bowman's capsule. Lumen is nothing but the cavity. That cavity is called as lumen. So whatever the cavity is present between the inner and outer layer of Bowman's capsule, here it is shown with this diagram, this one. See here, this region, this whatever is shown by this yellow colored curve, this is what cavity. Okay, so this cavity is present between this inner visceral layer, which shows the spatial type of cells called as podocytes, whereas the outer layer, which is made up of simple epithelial cells, and the space present between the outer parietal layer and inner visceral layer, that is called as Bowman's space or capsular region. Okay, and then it enters in the neck, and through the neck in the PCT, that is the proximal convoluted tubule. Now one more thing you have to remember, how this filtration helps, that the inner visceral layer shows the presence of, it forms, that also is made up of squamous type of epithelium, but that squamous type of epithelium is specialized into spatial type of cells called as podocytes. These are all podocytes, and in between the podocytes also there are small slits present, which helps in the filtration. These small slits are present, and even the podocytes are specialized, which create a pressure in between the blood capillaries, and that helps in the filtration, that is called as ultrafiltration. So this is all about the filtration in the glomerular region. Now what is that? That is first is called as glomerular hydrostatic pressure, that I told you already, and it filters out components of blood, and that is generally considered to be 60 mm of Hg, that I will tell you at the end of this part. Okay, what exactly that ultrafiltration process, what is the hydrostatic pressure that is created by glomerular area, that means glomerulus, that means blood vessels. Okay, then the second step, which is before that, I'll tell you what the ultrafiltration, that ultrafiltrate or the filtrate contains, or this is also called as glomerular filtrate or ultrafiltrate, or even that is called by the name called as primary urine. The filtrate which is formed, by the filtration of these blood vessels in the glomerular region, that is called as primary urine, or that is also called as glomerular filtrate or ultrafiltrate. So blood is filtered through these fine slits, as I told you, which are present in between these podocyte cells or podocytes. Even fenestrations are present. Fenestrations are also the same similar type of structures present over there, so which help in the filtration. Then this filtrate, what it contains then? It contains almost or everything except large sized molecules. Everything is filtered through these glomerular region, through this glomerulus. What that contains then? Except protein and the blood cells and the platelets, everything is filtered. So that filtrate, that ultrafiltrate or glomerular filtrate or primary urine, that is called as, that contains almost all the constituents which are of smaller size, all the molecules except the large size molecules like proteins. So that is also called as deproteinized plasma. So that filtrate is called as plasma, but the proteins are not present because proteins are large size particles. But what that plasma contains, all the small size molecules like glucose, amino acids, some salts, etc. are present. So first step is what? Glomerular filtration. Now the second step in the formation of urine is selective reabsorption. So I think you have understood that where the glomerular filtration takes place, in the Bowman's capsule, and then in the capsular space, through the capsular space, that filtrate or primary urine enters in the next region, that is called as PCT, that is proximal convoluted tubule, where this tubular structure, that is the renal tubule, in this region PCT is highly coiled, and because of highly coiled nature, what happens? The flow of that filtrate becomes very slow. It becomes very slow, and in this region, where because of this slow flow of filtrate, and also one more thing that is very important about this PCT is the endothelial or the inner linings of this PCT. They are made up of epithelial cells which are very specialized with the presence of microvilli, or you can say brush border like cells are present. Microvilli are nothing but these are the outgrowths, very small outgrowths which look like hair like structures, and they form, give the appearance of brush border. And because of that structure of epithelial cells present in the inner linings of the PCT, they increase the area of absorption. So these tubular epithelial cells in the different segments of nephron reabsorb certain substances of the glomerular filtrate, either by active or by passive mechanism. Now this selective reabsorption depends, where it may be passive. For example, passive, generally water. In case of water, water is reabsorbed passively. Passive, as I told you, energy is not required, not consumed, whereas active means where ATP is utilized. Now what generally in case of water which contains water in this filtrate, that can be reabsorbed by the walls of this PCT. As I told you, the structure of PCT, that the inner lining of the PCT, they show the presence of specialized cells, epithelial cells, that shows the presence of microvilli, which give the appearance of like brush. So these help in the absorption of the substances. So whatever the filtrate is passing through this tubular region, PCT, maximum filtrate, maximum contents of the filtrate, they are reabsorbed by the cells of PCT, either actively or passively. Water can be absorbed passively, where simple diffusion, you know how it takes place. From diffusion is from higher concentration to the lower concentration. So some substances with high concentration, they which are present in the filtrate, they can be absorbed by the walls of the PCT by simple diffusion process. Then in the selective reabsorption, here you can see this coiled structure of PCT, highly coiled structure. So the flow of filtrate becomes very slow, and during that time, as the epithelial cells are showing the presence of spatial type of structures called microvilli, these microvilli help in the absorption, they increase the area of absorption, and so that's why maximum amount of the filtrate, almost 85% of the filtrate is reabsorbed in this region of PCT, and remaining, that is loop of Henle's loop. So here in this highly coiled structure of PCT, the flow of filtrate becomes very slow, as well as maximum reabsorption, that is almost 85% of the material, the molecules, they are reabsorbed by the epithelial microvilli of the PCT. So this is very important part of selective reabsorption. Selective because everything cannot be reabsorbed, but selectively, so only some substances, for example water is reabsorbed, then some molecules, some salts, that also can be reabsorbed, like NaCl, etc. So this is very important part, that is PCT as well as loop of Henle, both the structures are involved in the selective reabsorption. Now here this is the PCT now, which continues in the loop of Henle. Now if you see this diagram, the loop of Henle, which shows this downwardly directed limb, that is called a descending limb, and upwardly directed limb, that is called as ascending limb. Now you can see the difference in the diameter of descending and ascending limb. If you see, the descending limb is with the, this is thin wall with the less diameter, whereas ascending limb, the proximal part of the ascending limb is with the low diameter, less diameter, but when as it passes, goes towards the DCT, that diameter increases, the thickness increases, and that diameter of this also of the ascending limb increases. Now how this loop of Henle helps in the reabsorption, try to understand. Now in this loop of Henle, the area which is of the ascending limb, that ascending limb of loop of Henle is having the ability of reabsorption of water. That one thing you remember, that is this region is permeable to the water, and the other limb, that is ascending limb of loop of Henle, it is impermeable to water. Now so when this loop of Henle descending limb is permeable to water, so what happens then? Maximum water is absorbed by the walls of inner lining walls of this loop of Henle of descending limb, and as the maximum water from this region, as it is permeable to water, this wall, water is reabsorbed. So what happens? Maximum water is reabsorbed by the cells. So what happens then? Concentration of the filtrate is increased, and then that means the urine, what we call as primary urine, that becomes concentrated. Then when it continues and enters the ascending limb through getting the U-turn, now this area, that is the ascending limb, is impermeable to water, but it is permeable to electrolytes. Electrolytes means all these ions. So this PCT as well as especially loop of Henle is playing very important role in the maintaining of hydrogen ion concentration, that is pH, as well as ion concentration, whether NaCl, etc. So when this primary urine, which we call, or the filtrate or ultrafiltrate, is passing through the ascending limb, first from the proximal part, that is narrow, and then to the thicker region which of this ascending limb, then what happens during this difference of permeability of water and impermeability of water of the descending and ascending limb of loop of Henle, what happens then? NaCl, such type of ions, they are removed out of this, they are thrown out of this tubular region. Okay, and so this is what happens during the change or when the filtrate moves for descending loop, taking the U-turn through the proximal part of ascending loop to the distal thicker region of the ascending limb, ascending limb of loop of Henle. So this is what helping in maintaining the pH as well as ionic concentration, ionic balance of the urine or the substances in the filtrate, that is the renal, as well as the remaining, this region is covered by the blood vessels, that is called as, these are, that is what, these are tubular regions or peritubular region. That means loop of Henle is covered by the blood vessels, highly called blood vessels, that is peritubular region. And as I told you that loop of Henle, this part is covered by blood vessels called as vasa recta. Okay, so you remember the things that in the selective reabsorption, which takes place mainly in the PCT as well as loop of Henle, that is the first descending and then ascending limb. So the next part of this formation of urine is tubular secretion. Now, the third step in the formation of urine is tubular secretion, and during this process, tubular cells secrete substances like ammonia, hydrogen ions, potassium ions in this region, which is shown by this yellow arrows. These yellow arrows, that means this is the part of DCT. Try to understand. This is a part of DCT. That means what? Distal convoluted tubule. Now we studied first two steps, that is glomerular filtration taking place in Bowman's capsule, and then next is the selective reabsorption or tubular reabsorption, which is taking place in PCT and loop of Henle. And the third step of urine formation, the last step, that is called as tubular secretion, that is taking place in the DCT region of renal tubules, that means distal convoluted tubule, as well as to some extent in the DC, that is the CT, that is the collecting duct. Next to that is collecting duct. So during this process of tubular secretion, what happens then? Ammonia, hydrogen ions, potassium ions, they are secreted by the surrounding blood vessels. Now you can see this region, that region is covered by highly coiled or blood vessels. These are called as peritubular blood vessels or peritubular capillaries, or the region where they secrete these ions to maintain the ionic balance, like H plus ions and K plus ions for maintaining the pH and ion concentration. So tubular secretion takes place in the DCT region, where it helps in maintenance of ionic and acid base balance of the fluids. That is very, very important. Now again we'll repeat this process, how this mechanism of selective reabsorption and secretion takes place in different parts of the nephron. Now as you know that this is the first step, that is capsular space is the region of the first step of process of urine formation, that is glomerular filtration. Then these are the cells of PCT. This is when feed enters Bowman's capsule, enters the renal tubule. First it becomes narrow, that is called as neck, and then it enters PCT, and the cells, epithelial cells in this region, they are showing the borders, giving the appearance like brush border, and they are called as microvilli, which increase the area of absorption. So brush border of cuboidal epithelium of PCT increases the surface area for reabsorption. So what is reabsorbed then? These are the substances which are reabsorbed in the PCT region, like glucose, amino acids, Na ions, Cl ions, then water. All these are reabsorbed either passively or actively. Passively, for example, by osmosis, very easily, passively, or by diffusion, this part takes place. So without using much energy, this takes place, then that is considered to be passive. And when it is reabsorption is using requiring energy in the form of ATP, then that is called as active type of reabsorption. Now, loop of Henle is the part where these two limbs, descending and ascending, also are playing a significant role in maintaining the osmolarity of the fluid. That means osmolarity is the concentration. As I told you, concentration of the solute. Solute is what which is passing through this tubule. As I told you, this descending limb is permeable to water, but whereas ascending limb is impermeable to water. So maximum reabsorption of the water takes place here, and when water is reabsorbed by the cells, it is passed to the blood tissue. So what happens then? The concentration of the solute, that is the urine, is increased as water is removed from this region, from the filtrate. So concentration of the urine is increased, and as it passes through the ascending limb, again NaCl, these are passed out. So in this region, it helps in maintaining the concentration. So what osmolarity is nothing but the number of osmoles of solute per liter of solution. That is nothing but the solute concentration is measured in the form of osmolarity. Solute concentration here, you just remember that is in the urine, she call is that primary urine, which is passing through this loop of Henle. Because of the different nature of permeability for water, that is of descending and ascending limb, it helps in maintaining the osmotic balance, that is the osmolarity or urine concentration. Then as it maximum water is permeable, so that is reabsorbed, and so this becomes shows the urine in more concentrated form. Then this limb, as I told you, it is impermeable to electrolyte, descending limb, but permeable to water, but exactly opposite about this ascending limb. So reabsorption of water continues as the filtrate moves along the descending limb by passive transport. Now, this is what, again, I had shown with the help of this diagram, that NaCl is removed out, or it gets diffused from the descending limb because it is permeable to the electrolytes. So, this ascending limb is permeable to the electrolytes but not to the water. So, NaCl, so such ions are diffused from the ascending limb of loop of Henle, and ascending limb is impermeable to water. So remember these 2 points, very important in maintaining the pH as well as ionic balance of the filtrate, or you can say urine. Now the last part, that is DCT and then CT, when from here maximum secretion takes place, where in the distal convoluted tubule secretion of the ions like Na and as well as K plus etc. is taking place, as well as this loop DCT is also meant for the reabsorption of water as and when required, but the cells present in this region are not showing presence of any microvilli, so they are not directly involved in the much reabsorption. As and when required, water can be reabsorbed by the cells of DCT, and that also is controlled by ADH, that is antidiuretic hormone secreted by pituitary glands. So this DCT here, reabsorption is conditional, that is reabsorption of water is conditional, as the cells are of shorter size and they are not showing any presence of microvilli. This area is not directly involved in the reabsorption, but as and when required, this area of DCT as well as CT, that is the collecting duct, that reabsorbs water and to some extent ions from the surrounding capillary region, that is the tissue, blood tissue. Now, the next is, so as I told you that ADH plays is a very important, ADH, that is anti-diuretic hormone, which is secreted by the pituitary glands, and it is conditional in the sense, if the urine becomes more dilute according to the nature of the urine or the content of the urine, that release or reabsorption or secretion that of this water in the DCT region, that is controlled by the secretion of ADH. So ADH is the antidiuretic hormone which is secreted by the pituitary gland cells. So last part of DCT, where these ions like K plus, H plus, NH3, etc. can be secreted. So the secretion, reabsorption of this HCO3 and selective secretion of H plus, K plus ions, NH3 into DCT from peritubular region or peritubular network via cortical interstitium. That, just remember that the entire area is covered by the peritubular region. That means all the blood vessels are covering this area. So from the blood itself, as and when required, the selective reabsorption or secretion of such ions is done to maintain the pH of the cell, pH of the urine, as well as ionic concentration of this. So pH and sodium potassium balance in blood also can be maintained with this selective reabsorption as well as secretion. Now the last part of this nephron, which is called as CD, that is collecting duct, it carries the filtrate from the DCT region, where up to this the entire area is present in the medullary region, which it comes here to the, from the medullary region it comes to the innermost interior, that is the innermost part of kidney, that is called as pelvis. So from this pelvic region through the ureters, then which enters the ureter through from the pelvis region, and then it is carried to the, through this collecting duct. This collecting duct from this enters the renal pelvis, that is the pelvic region of the kidney, and from this region where the ureters are present, through the ureters it is passed to the bladder, that is urinary bladder. Okay, so considerable amount of water could be reabsorbed from this region to produce concentrated urine. So maximum water is reabsorbed so that the urine concentration is more, and it is very important in keeping the pH. Now one more thing, additional thing you need to remember, that urea, which urine contains, which we are calling now urine, that contains urea. So some amount of urea also can be reabsorbed by the surrounding cells of blood cell, that is the tubular, peritubular region. So that secretion and reabsorption as and when required, that takes place in this CT region, that is the collecting duct region, and that helps in maintaining the pH, because the urine is highly alkaline, more alkaline, but because of this selective secretion of H plus and K plus ions, then it maintains the ionic balance of the blood and also pH. This is very important, and that is controlled by ADH, so that you remember that the urine in the CD is hypertonic to the plasma of blood. So this region is very important, that this DCT region is highly coiled or covered by this peritubular region of blood vessels, and so this is the collecting duct. So this is what all the steps taking place in the region of nephron: cortical region, medullary region, and then to the pelvic region. Through the pelvis it enters the structure called as ureter, the which from where it is passed to the urinary bladder. So remember that urinary secretion is nothing but the glomerular filtration minus tubular reabsorption plus tubular secretion. So whatever it is shown, that is in a very small or in a short way, it is shown in this uh, equation, you can say. That is urinary excretion, that means urine, how it is formed through glomerular filtration minus tubular reabsorption and tubular secretion, that I will explain here with the help of your textbook later. Now, whatever the substances are reabsorbed or they are formed here, they are based on their necessity of reabsorption, and accordingly they are categorized into three types, that is high threshold substances, low threshold substances, and a threshold substances. Now what are these? First, then, as I told you, that the filtrate contains all the molecules except, means all the small size molecules except the large size like proteins. So what they contain? Glucose, vitamins, amino acids, salts, and many more. But these substances, they are very essential for the body. And that's why whatever the filtrate content, that is the primary filtrate or primary urine or ultrafiltrate contains, all these are required. So they are again reabsorbed. So what are these? Highly efficiently reabsorbed, which are very essential for the body. They are glucose, vitamins, amino acids, and some salts. So they are called as high threshold substances. And second is low threshold substances, which are not essential for the body, like urea, uric acid. They are passed out of the body, thrown out of the body, but to maintain the ionic balance, sometimes they are absorbed in a very little amount. So they are called as low threshold substances, which are not essential for the body and absorbed in a very little amount. And the third category, that is called as threshold substances, is which are not essential for the body at all, and they are not reabsorbed at all. For example, creatinine, that can not be reabsorbed, that is not reabsorbed, but it is thrown out through that filtrate itself called as urine. So these are, this is what a threshold means. Not threshold, that means not essential or not required for the body. So glucose as high threshold, urea as low threshold, and creatinine as no threshold or a threshold substance. Now the last part, which is considered to be mechanism of concentration of the filtrate or counter current mechanism. I will explain this in a very simple way. Just try to understand. As I already explained, that the loop of Henle, which is having the two limbs, one is descending and one is ascending, which are having the playing the very important role. And these are, that loop of Henle is covered by vasa recta. That means all the blood vessels which are covering the loop of Henle, they are called as vasa recta, and they are playing a very important role, significant role in the production of concentrated urine. And this is very important, that this is what called as vasa recta. As I told, loop of Henle is covered by this peritubular region, and that's why this is called as vasa recta. So the flow of renal filtrate in the limbs, two limbs of loop of Henle, is in opposite direction, and that is what counter is what? Opposite. This is the loop of Henle, the entire one. This is ascending limb, this is descending limb, and this is ascending limb, and both are in the opposite direction. They are running in the opposite direction. That's why this is called as counter current mechanism. So the flow of urine in is the opposite direction. The same way, the blood vessels which are covering the loop of Henle, that is the vasa recta, and all this network of blood vessels, they also are running in opposite direction. One is descending and the other is ascending. Here also this is the counter current mechanism or the pattern. That means the flow of blood through these two limbs of vasa recta is also flowing in the opposite direction. This is descending direction, descending limb of the this peritubular region, that is the vasa recta, and this is the ascending limb. So both are opposite. This is the descending limb of the renal tubule, that is the loop of Henle, whereas this is the ascending limb of the vasa recta. So both are running in the opposite direction, and that helps in the counter current mechanism where the urine is made concentrated. Because of this counter current mechanism, maximum reabsorption takes place, and as both the limbs of loop of Henle and vasa recta also, they are in opposite direction. That is what counter current pattern. This is called as counter current pattern, and which help in maintaining the osmolarity, that means the solute concentration, which is generally 300 per liter and about 1200 per liter in the cortex and medullary region respectively. So human kidneys produce urine nearly 4 times concentrated than the initial filtrate form. Where is the initial filtrate formed? In the capsular region of Bowman's capsule, which from where 85 to 99% of the filtrate is reabsorbed by the different regions of the nephron. Now, just this only we discussed right now. So, on an average, 1100 to 1200 ml of blood is subjected to filtration by the kidneys per minute, which constitute roughly 1/5 of the blood pumped out by each ventricle of the heart in a minute. And what is the GFR? That is glomerular filtration rate, which where the amount of filtrate formed by both the kidneys per minute is called as glomerular filtration rate, and it is approximately 125 ml per minute, that is 180 liter per day, that is GFR, glomerular filtration rate. So that much of blood is blood is filtered. So the kidney normally excretes only 1 to 1.5 liter of the urine in 24 hours of period. Although about 180 liter of glomerular ultrafiltration is produced each day, nearly 99% of the filtrate is reabsorbed by the renal tubules, and this process is called as reabsorption or selective reabsorption. This is what about the formation of urine. Now, we will in the last 6 minutes we will just revise a little